temperature, and always write down the last one as water. So the water is the least important limiting factor. So I wanted to write it like the last one separately. But these three, like the main three factors that going to limit the rate of photosynthesis. Water is something different. So I'm going to take the first two. The main inputs, light for the photosynthesis, and the carbon dioxide concentration for the light independent reaction. So let's take the light intensity first. Write down, out of the two reactions, light dependent, light independent, which one affects the, by changing the light intensity? And at the same time, you're going to plot the graph. This is one of the dot points in the study design, and you have to know this. Can you label the axes? So it's the same thing, right? So whatever you have the on the x-axis is the independent variable. So what we are changing is the light. And because of the light, what's going to change? The dependent variable is the rate of photosynthesis. So if I change the light, the main state that's going to change is the affected, going to be the light dependent reaction. Write down the light dependent reaction. So we are using water molecules. To split the water molecules, I'm using light. To produce energy, right? So write down the energy. Just ignore the oxygen. We don't need oxygen now. We are only looking at the main inputs and output. So if I use water, when I split water using the light, we're going to produce energy, right? ATP and NADPH. So higher the light intensity, higher the energy going to get to split water molecules. So if you give like a lot of light, lots of water molecules going to be splitted, and we're going to produce lots of energy, which can go and power the light dependent reaction, independent reaction. So light independent is mainly carbon dioxide into glucose. Okay, so if I have a lot of light, I can split a lot of water molecules, producing a lot of energy. So you can write down both energy types, which can increase the amount of glucose produced. Right, so this is what I'm going to write down. And then we're going to just plot it using this graph. So I'm going to plot the graph. Can you describe why the shape is going to be like this? So this is how the light intensity graph going to look like. If I increase the light, this is how the rate of photosynthesis is going to be affected. Write down why the rate of the reaction going to go up from A to B. And same as the enzyme graph, there's going to be limiting factors that are going to limit the rate of photosynthesis, right? So these are the limiting factors other than the light intensity. So we have plenty of light intensity available from point B onwards, but other factors going to be limiting from B to C. Can you write down the other type of limiting factors? So from B to C, the constant photosynthesis, the glucose amount is due to the limiting factors, right? I will write it down. Now, if you can write down why the rate of the reaction going to increase from A to B, and why the rate of the reaction is constant from B to C. The explanation for B to C is the limiting factor, right? There are lots of limiting factors. Write down a couple of limiting factors if you can think something. You can write down the A to B explanation is this, right? So otherwise I can rewrite this. So if you read the x-axis from A to B, you can see the light intensity just gradually increase, right? So rate of photosynthesis is going to increase up to the point B, right? So this is because the light absorbing pigments like chlorophyll absorb a lot of light. So that means we have a lot of light available to split more water. So this creates more ATP and NADPH. So this energy can be used to produce more glucose, right? That's what I wrote down earlier. Exactly the same thing I'm rewriting it. This is a dot point. I have to know all the limiting factors. So as the light intensity gradually increase, the rate of the reaction also going to increase up to the point P. So this is the explanation for this increase. But after the B, there are limiting factors other than the light intensity. As an example, carbon dioxide concentration might not be enough, right? So even we have like lots and lots of light available, other factors like carbon dioxide going to be a limiting factor. Is this clear? It's the same as like the enzymes and the substrates and those graphs we drew earlier, right? The enzyme graphs. So if this is clear, can you identify a limiting factor that limit the rate of reaction? Highlight this part. Huh? So when it comes to the VCA exam questions, they can ask questions like this. So if I highlight any part between A to B, or just highlight just the line only. So can you identify a limiting factor that will limit the rate of the reaction from A to B? Write it down. Can you identify a limiting factor why the rate of the reaction is not up to the maximum from A to B? B is the maximum rate, right? But if you think about somewhere here, so definitely this point is less than the point B. The question I'm asking is, why this point here, the rate of the reaction is less than point B? Write down a limiting factor that limit this rate of reaction in this point, whatever the point, if I say point X. So this is before the saturation point, right? So this is the saturation point. We have like the maximum light intensity, but there's going to be other limiting factors. Send me the answer for this one. Before point B, why the rate of the reaction is less? Someone please send me the answer. So after the point B, it is like something else, right? It could be the carbon dioxide concentration. But if you consider something before this maximum point, point B, it has to be the light intensity. Is this clear? Because if you think about here, this point here, so the light intensity maximum is somewhere here, right? So if I say 100, 100 is the maximum light intensity, but if you think about something before, like 20, 20 is not enough to increase the rate of photosynthesis. So anything before the saturation point or the maximum rate of reaction, the limiting factor needs to be something on the x-axis. It's always the x-axis. Right? So in this example, light intensity is limited. So the rate of the reaction is less from A to B. But B to C, you can see other factors like carbon dioxide limiting. So that's why the rate of the reaction is constant. So I'm going to write down again, A to B, the limiting factor is light. So B to C, it could be any other factor other than the x-axis. Is this clear? I'm just saying the same thing. So if you understand this, can you do the similar one for the carbon dioxide? Draw the graph. You have to show exactly the same graph. Only thing is you're changing the x-axis for carbon dioxide. So we did the light intensity. Can you draw the graph for the carbon dioxide and write down the reasons why the rate of the reaction is increasing from A to B and B to C? Identify the limiting factors for A to B and B to C. Exactly the same thing. So you have to explain why the rate of the reaction goes up from A to B and write down the limiting factor as well for A to B. And then write down why the rate of the reaction is constant from B to C. You might get like these kind of graphs. You have to just describe both, right? So why the increase and then why it is constant. So the state that's going to be affected is the light independent reaction. Because carbon dioxide is the substrate for the Calvin cycle. Higher the number of carbon dioxide molecules, higher the number of glucose that's going to be produced. So if you consider anything before the saturation level, the maximum photosynthesis rate B, what's going to limit the rate of photosynthesis is the carbon dioxide concentration. This is something you have to know, right? Because carbon dioxide is not to the maximum, right? So the maximum is somewhere here. So if I say this value is 50, so you have to have 50 carbon dioxide molecules to get the maximum rate of photosynthesis. Anything before needs to be less, right? So limiting factor that's going to limit the rate of reaction anywhere in between A to B has to be always the x-axis. This is something I always see in beta exams. But after B to C, it is constant. We have enough carbon dioxide, but other factors like light intensity could be limiting. Right, so I'm going to write down light. Or you can say not enough chlorophyll pigments available to capture the sunlight. Don't write water. Plants can have enough water always. Don't write like water as a limiting factor. If you don't have anything, you can write down. Otherwise, just write down something else other than water. Is this clear? These are dot points. You have to know the graphs. You have to know the explanation, right? So carbon dioxide is the input for the Calvin cycle. Higher the number of carbon dioxide, higher the number of glucose we can produce. But the rate of the reaction is going to just increase up to a maximum point, and it's going to just go constant because other factors like light or the number of chlorophyll are going to be limiting factors. Let's do water. No. Temperature. Is this clear? If you have any questions, just ask. So I did the light, I did carbon dioxide. Let's do the temperature. So temperature is exactly the same thing we did for the enzymes, right? So can you plot the graph for the enzymes? The normal enzyme and the rate of the reaction graph. label the axes. What we are changing is the enzyme. Because of the enzyme, how the rate of the photosynthesis is going to change. Right? Or you can say the amount of glucose produced. Plot the graph. The rate of the reaction is going to increase up to the maximum and it's going to just come down. Okay, can you write down which state they're going to be affected by this enzyme concentration? Write down the main enzyme that's going to lead to this glucose production. The main enzyme. Rubisco, right? Rubisco is the one actually produce the glucose. So Rubisco is the main photosynthetic enzyme. Write down the plants that needs to have this Rubisco in which one? C3, C4, OCAM, or all. So if you're considering an enzyme, it needs to be Rubisco always, right? For photosynthesis, it's the Rubisco. Because Rubisco is the one that's going to produce the glucose, right? Calvin cycle is going to be using Rubisco only. So it needs to be present in every single plant, C3, C4, OCAM, photosynthesis. Write down another enzyme other than the Rubisco. So it's not present in every single plant, right? So it's going to be an adaptation to live in hot and dry environment. So this enzyme is the PEP carboxylase. But PEP carboxylase not present in every single plant like the Rubisco. So if you're like mentioning an enzyme, it needs to be always Rubisco, right? So PEP carboxylase present in C4 and CAM. Because they are the ones that are doing the initial carbon fixation. So always highlight Rubisco, not the PEP carboxylase. I don't know why I wrote enzyme instead of temperature. So the third factor is the temperature, but it's going to affect the enzymes, right? So temperature is the factor, but the temperature is going to affect the enzymes, right? So enzymes are affected by the temperature. So the main enzyme needed for the photosynthesis is the Rubisco. Rubisco is going to affect the light independent or the Calvin cycle. Okay, can you explain? I'm going to write down the keywords. Can you all explain why the rate of the reaction is going to be less at lower temperatures, lower than the optimum temperature? So if I say somewhere around 10 degrees, why the rate of photosynthesis is low? Give me a minute. Huh? So this is the why the rate of the reaction is low. Can you use these keywords? Remember here the main substrate is the RUBP, the 5-carbon RUBP. Use those terms. And can you include the kinetic energy? And the number of collisions? And then talk about the glucose formation. Start with the temperature being low. So if the temperature is low, the kinetic energy between the Rubisco and the substrate is going to be low. Right, so the Rubisco and the substrates, they have less kinetic energy, so they are moving slowly. So because of less movements, number of collisions between the Rubisco active site and the substrate going to be less. And this is going to lower the glucose formation. Right? Less glucose means slow rate of photosynthesis. Is this clear? Can you highlight the keywords? You have to mention the kinetic energy. And then you have to mention the collisions between the active sites and the substrates, right? You have to use the keywords. Substrate is the main substrate is RUBP. The main enzyme is the Rubisco's active site, needs to have the collisions. So write down why the rate of the reaction is going to slow down if you increase the temperature beyond the optimum temperature, right? I highlighted them in green. Use the term Rubisco's active site, talk about what happened to the active site. And the second one is, the main thing is the denaturation. Use the two keywords and write down the answer. So if you remember the bonds, you can talk about the bonds, right? So Rubisco is an enzyme. They need to have the hydrogen sulfur bonds, but very high temperature is going to break down the hydrogen sulfur bonds. So because of the bonds breaking down, hydrogen sulfur, the Rubisco is going to lose its active site. No Rubisco's active site. So without the active site, the substrate can't bind. Somewhere I don't denaturation. So if I go beyond the optimum temperature, the enzyme Rubisco going to denature, because high temperature is going to break down hydrogen sulfur bonds. So without the hydrogen and sulfur bonds, no active site. Rubisco going to lose its active site, so substrate can't bind, no product formation. Somehow write down the two keywords. So these are the three main factors. You have to know the graph and the explanation. Is this clear? So shall we do the last one? Anyone writing this down? I can't see most of you. Okay, so when we did the last year's old study design, they included only these three factors, right? Because these are the main three factors. But I don't know why they have included water as well this year. But just leave it as the last one. Huh? Water is not like a main factor. So let's do water. So last one, the factor that limits the rate of photosynthesis is the availability of water. Okay, so if water is like a limiting factor, it's going to affect the light dependent, right? Because light dependent reaction, the main input is water. So if I have light, I can use the light to split a lot of water molecules to produce energy for the light independent. Oops. Right, so theoretically, this is the explanation. Higher the water molecules, higher the energy we can produce. So more energy means more glucose can be produced. Right, so if you really want to know the explanation, this is it. But write down this one first. Write down how the water is going to come into the plant. We did this last year, right? You did the systems, you did the xylem phloem. How the plant's going to get water into the plant. So initially, water needs to come through the roots. And then it comes to the xylem. Remember xylem last year? And xylem transport water throughout the plant, right? But the initial structure that needs to absorb water are the roots, right? Xylem is just the tube transport water. But normally, plants need to have lots and lots of water inside to sustain the life. So as an example, for all the metabolic reactions, they need water. So they're going to have lots and lots of water stored. As an example, they have a vacuole. Inside the vacuole, they will have lots of water stored. So to sustain the plant life, the plant needs lots and lots of water. So compared to this amount, the amount we need for the photosynthesis is very, very small. So if I want to maintain a plant without killing it, I need to give lots of water. So the plant cells will become turgid. Turgid is the good condition. Otherwise, plants will wilt. Remember what the turgid is? That means outside water environment needs to be hypotonic. So normally the plant needs to have lots and lots of water inside. So this amount is very big compared to the amount we needed for the photosynthesis. So if in case a water is a limiting factor, the plant is going to die before it limits the photosynthesis. Right? Lot more water is needed to sustain the plant life than the amount needed for the photosynthesis. Is this clear? So that's why I don't write it as like a main factor. This is not like a main limiting factor. But water can be an indirect effect. So water not going to be like a direct limiting factor. But if the plants are under water stress, not having enough water inside, what happened to the plant stomata? So if this is like a cross section of a leaf, if the plant's under water stress without having water inside, they can't leave their stomata open because whatever the remaining water will evaporate through the stomata. Right? So if in case if the amount of water inside is decreasing inside the plant, they need to close their stomata. Right? So because of the water stress, they might need to close their stomata. So this is going to limit the amount of carbon dioxide needed for the photosynthesis. So the plant will uptake all the available carbon dioxide. So the main thing is the plant won't have enough carbon dioxide to produce glucose, right? So if it is like C4 CAM plant, it doesn't have enough carbon dioxide to produce the glucose. If it is a C3 plant, it might undergo the photorespiration. Right? So water stress is going to pose an indirect threat by cutting down the amount of carbon dioxide if the plant closes their stomata. So decrease the carbon dioxide means you're decreasing the glucose. Right? So decreasing glucose means rate of photosynthesis is decreasing. Is this clear? So if you really have to explain the water availability as a limiting factor, you can talk about this effect. If the plant is under water stress, if they close their stomata, they will have a problem to uptake carbon dioxide needed for the glucose production. Is this clear? So there's a small part on photosynthesis. I will do this later. Otherwise, I'll just send you the note. You can read it. So there's a CRISPR-Cas9 part again. How the CRISPR-Cas9 can be used to improve the crop productivity. I'll do this part later. Because some of the students doing the cellular respiration sacks now, I'm going to start the cellular respiration. I'm going to finish this lesson next week. That means I'm going to finish photosynthesis. I finished already. So we went through the C3, C4 and CAM photosynthesis and whatever the limiting factors. Before that, all the cycles, the light dependent, light dependent, limiting factors, I just went through. And then I'm going to do the cellular respiration now. Right, so I'm going to finish this next week. I'm going to give the test on 13th. If you haven't done this at school, you will do this very fast, right? So this is the next topic. It's a very big part we haven't done yet, immunity. It used to be in unit 3, it's very, very big, right? So before we start doing immunity, just do whatever you know. Go through the photosynthesis, cellular respiration. These are very, very easy topics compared to the other stuff. So I'm giving like enough time, please go through the content. Okay, write down the topic cellular respiration. So this is exactly the same thing we did last year, right? There's no change, exactly the same thing. So before I do the cellular respiration, can you draw a big mitochondrion like this? Can you, if you remember, can you draw the matrix, inner membrane, outer membrane, and cristae? Don't draw 3 membranes. Mitochondria has only 2 membranes. Do not draw 3 membranes. We have students doing like this. So if you do like that, I don't know what this is, but 1, 2, 3. There are 3 membranes. And then write down the function of the mitochondrion, main purpose of the mitochondria. Okay, I'm going to draw it. Outer membrane is the flat membrane. Don't do microscopic ones. You might need to know how to draw this. So draw like a bigger one. Outer membrane is flat. The inner membrane, there are cellular respiration reactions happening on the membrane. So it needs to have a long surface area than the outer membrane. So we need to make the inner membrane longer than the outer membrane. So for that, we're going to have these foldings. So if I straighten the inner membrane definitely longer because of the presence of these parts. Inner membrane is not flat. So inner membrane going to have the foldings like this. Right, you can highlight all of them if you want. So these are the cristae. Right, so inner membrane foldings are the cristae. And the last remaining one is the matrix. Matrix is the liquid. Right, so the cytosol-like liquid inside the mitochondria is the matrix. Right, so you can just color it when you don't have anything. So this is the matrix. So some of the cellular respiration reactions going to happen inside the cristae. Some of the reactions will take place in the matrix. So you have to know what the matrix and the cristae are. Right, so the most important two parts for the cellular respiration is the matrix and the cristae. The mitochondria's main function is this is the site of cellular respiration. Or you can say this is the site of ATP production. Right, so can you write down the cellular respiration balance equation? Is this done? Everyone finish, yeah? Okay, write down the balance equation. This is just the opposite of photosynthesis, right? Don't do silly mistakes, huh? Don't swap the photosynthesis and cellular respiration. So cellular respiration means you're breaking down glucose. Glucose is a 6-carbon compound like this. We are going to undergo a catabolic hexagonic reaction and break down the 6-carbon compound until it becomes 1 carbon dioxide. This is not the balance equation, no? But the main thing is to break down glucose until it becomes 1 carbon dioxide. So you're going from 6 carbons to 1 carbon. The main purpose is because this is going from 6 to 1, it is releasing lots and lots of energy, which is going to be trapped as the main energy source ATP. So this is the reaction that's going to produce all the ATP for the entire cell. Right, so cellular respiration is basically producing ATP. How are we going to do it? By breaking down a big compound until it becomes one carbon dioxide. Let's write down the balance equation. So the main purpose is to break down glucose until it becomes carbon dioxide. But we're going to use oxygen as well to produce water. Right, so I just separate out the two inputs. The glucose will become carbon dioxide. So can you please change the oxygen? And oxygen is the one that's going to become water. So I can do the same thing as I did for the photosynthesis, right? So I can use radioactive compounds, I can label them and ask, if I add radioactive oxygen, what happens to the oxygen? You have to know this is the oxygen that end up in the water molecule. I'll explain the stages, but change the colors. Huh? So glucose will become carbon dioxide. Oxygen will become water. Balance it. Don't write unbalanced equations. Huh? No marks. So 6, 6, 6. Only thing is, don't write the light. Right, so this is the cellular respiration balance equation. Finish? They have finished? Okay, so I don't need the oxygen and water, right, until towards the end. So I'm going to just erase it. Cellular respiration, the main thing is to break down 6-carbon glucose until it becomes carbon dioxide, right? So every single living cell must undergo cellular respiration by breaking down 6-carbon glucose until 1 carbon dioxide, right? So this reaction, I said, going to release ATP as the main energy source. Can you write down the two main cells are the plant cells and the animal cells, right? We are not considering like the fungi and the protists. Okay. How the plants going to get their glucose for the cellular respiration, right? So plants or animals, if they are living cells, if they are living organisms, they must produce their ATP. Without ATP, plants can't survive, animals can't survive. So that means both of them, not only the animals, plants also must undergo cellular respiration. Plants can stop the photosynthesis, but they can't stop cellular respiration, right? So without respiration, there's no ATP. Without ATP, they can't survive, right? They can't fulfill their energy needs, they're going to die. How the plant's going to get their glucose? Yeah. How the plant's going to get their glucose? How the animals going to get their glucose? Animals include humans. Write down the source of glucose for the plants. What is the source of animals? The source of glucose for the animals or the humans? How are you going to give your body the glucose? So remember, not only the animals, plants also must undergo cellular respiration, right? So that's why we went to the photosynthesis. Photosynthesis is the output for this glucose. So plants are autotrophs, they can make their own glucose because they are autotrophs. I introduced these terms, now you can use them. But animals are heterotrophs, they can't make their own glucose. So what they're going to do is they're going to eat the glucose made by the plants. So if you eat like a starch, a big potato, so starch is made out of combining lots and lots of glucose. So when you're eating starch or a potato, you're eating like lots and lots of glucose bonded together, but you're eating like starch, a big carbohydrate. But when you consume this, so this is your body or animal's body, your digestive enzyme is going to break down the bonds and release glucose for the cellular respiration. Whether the cells can make their own glucose or they're going to get it from another source, the main input needs to be the glucose, right? So whatever the way, everyone must have glucose to undergo cellular respiration. Is this clear? Right, so cellular respiration is the process of producing ATP. To produce ATP, we have to have glucose. Plants make their own glucose, animals take whatever the autotrophs produce, right? And use the digestive enzymes to process the glucose. Okay, let's do the 3 stages. Right, you learned these 3 stages last year, exactly the same 3 stages. So I'm going to write down the 3 stages now. So can you draw like a big cell with the mitochondrion? So cellular respiration is, we are going to break down glucose, 6-carbon glucose, until it becomes 1-carbon carbon dioxide. Right, so this is an exergonic reaction going to release ATP. The first stage is we're going to break down the glucose in the cytosol. If you remember last year, the first stage of cellular respiration is the glycolysis. Write down the location. What is the location of the glycolysis? So it's not inside the mitochondrion yet, it's outside, right? So the location of the glycolysis is, glycolysis takes place in the cytosol. Right, so this is very important. The first stage of cellular respiration is outside, not in the mitochondria. And then write down the next two stages. I'm going to erase them. You're going to have to explain I'm going to explain glycolysis first. But the second stage is like the last year, it's the same Krebs cycle. If you remember, can you write down the location inside the mitochondria? So whatever the product that produced from the glycolysis is going to go into the mitochondria and undergo Krebs cycle in the matrix. And the third stage is electron transport chain. So electron transport chain going to be in this cristae. So when you're writing the locations, in 2016, they did not give marks for just writing matrix or cristae. Don't write just the matrix cristae, you're losing the mark. So it happened for this matrix because there might be another matrix somewhere. Right? So can you write down Krebs cycle going to take place in the mitochondrial matrix? Do not write just the matrix, you're losing a mark. Electron transport chain, instead of saying cristae, say mitochondrial cristae. Or just write it down, it's part of the mitochondrion. Right, like this. So I'm going to start breaking down the glucose. Glucose has 6 carbons. I'm going to use 3 stages to break down glucose until it becomes 1 carbon dioxide. Everyone finish? Okay, shall we start the glycolysis? Okay, let's start the glycolysis. So I'm going to start breaking down the glucose. So we're taking the glucose molecule, the 6 carbon glucose, and we're going to split the glucose to half. Write down the number of carbons in the product. You're just splitting the 6 carbon into half. So the product going to have 3 carbons each. Right? So like the same as the last year, when you split glucose in the glycolysis, the product is pyruvate. Right? You don't have to know the chemical formula or anything, just the pyruvate, and you must know it as a 3 carbon compound. So this is a big hexagonic reaction because I went from 6 carbon to 3 carbons. So if you break down something, always energy is the output. So the main energy source is the ATP. We are producing 2 ATPs here. Plus, there's going to be high energy protons. Can you write down somewhere? We are doing the cellular respiration. Can you write down the unloaded coenzymes we can use? What are the two coenzymes we can use in cellular respiration? We are not doing photosynthesis now, right? So when it comes to photosynthesis, we use NADP. That's the coenzyme. But we are doing cellular respiration. You can either use NAD, I'm writing the unloaded ones, right? So I can either use NAD or FAD. But for the glycolysis, we are only using NAD. Yeah? We're going to use the FAD in the Krebs cycle. So can you use NAD and go and grab the high energy proton? So this proton created by the splitting glucose, and then write down only the loaded version, right? Don't write the H plus, no need. Load the NAD with the high energy proton, and then write down inputs and outputs. What are the inputs and outputs of glycolysis? Right, very, very easy. There are 3 inputs and 3 outputs. There's no FADH2 here. Do not write FADH2 in glycolysis. It's just NADH. Okay, can you write down 3 inputs, 3 outputs? Start from the outputs, right? You can clearly see the outputs and then do the inputs. You don't have to know the NADH numbers, but you must know the ATPs. At the end, we are going to count them. Right? For glycolysis 2 ATP, you must remember the number. It's exactly the same as last year. Right, so the 3 outputs are, you have pyruvate. So pyruvate came from the breakdown of the glucose. Don't write all over the place. Huh? Pyruvate produced by the splitting glucose, and ATP and NADH. Right? Write the unloaded versions. When you're writing the ATP here, you don't have to write the numbers. Right? So only thing is we're going to count them. So when you're writing the main thing, main reaction, write down the ATPs. This is it. Inputs, outputs. These are dot points. You have to know this. Okay, so cellular respiration means we are breaking down glucose until we produce one carbon dioxide, right? So what I have done so far is I have used the glycolysis and split the glucose into half. So I have now 3 carbon pyruvate. Right, so during the glycolysis, we have half the glucose molecule. We have a 3 carbon compound, which can be further break down in Krebs cycle until it becomes 1 carbon dioxide. Right, so I'm going to use the whatever the product here, the pyruvate, the main carbon source, and we are going to go to the Krebs cycle to further break this down until we produce carbon dioxide. Right, so every single step we are going to collect the ATP. So let's take the pyruvate, we are going into the mitochondrion, right? So let's go to the mitochondrion matrix. So I'm taking the pyruvate towards the mitochondrion matrix, right? So still we are in the cytosol. So let's go to the mitochondrion matrix. Right, and then we're going to break down the pyruvate until it becomes carbon dioxide. Okay. So can you write down? So I took the pyruvate into the mitochondrion matrix, but pyruvate is going to break down. So we are going from 3 carbon and we're going to initially break down the pyruvate into a 2 carbon product. Right, some of the textbooks use this compound, that's why I'm introducing this. Write down pyruvate gonna break down into the acetyl coenzyme A. This is a 2 carbon product. You can say acetyl CoA, right? So inside the mitochondrial matrix, the pyruvate, the 3 carbon compound will break down into another compound, acetyl CoA. And acetyl CoA is the one that undergoes the Krebs cycle. Can you draw like a circle, a clockwise circle, and write down inside, this is the Krebs cycle. So what actually enters into the Krebs cycle is the acetyl coenzyme A. But if your teacher doesn't mention this, you can drop this and say pyruvate as the input of the Krebs cycle. Right? But this is even in the high level book, the Pearson book. So your teacher might just include this, but otherwise just forget it and then say pyruvate enters into the Krebs cycle and undergo the breakdown. Right? Either one is correct. When I'm writing inputs and outputs, I'm going to drop the acetyl coenzyme A. We're going to write down pyruvate as the initial input. But you can just remember there is something called acetyl coenzyme A. This is the actual product that will undergo the Krebs cycle. So if I say acetyl CoA as the input of Krebs cycle, anything happened before needs to be pre-Krebs cycle. Right? So write down, this is the pre-Krebs cycle. Is this clear? If the Krebs cycle starts from this, acetyl CoA breaking down in Krebs cycle, Whatever happened before needs to be before the Krebs cycle. So write it down, it's the pre-Krebs cycle. Justin Kirat, you there? Okay. So let's write down the, otherwise you can just drop this, huh? Just write down pyruvate as the input of Krebs cycle. So after the Krebs cycle, during the Krebs cycle, we are going to break down all the carbons and going to release the final output, carbon dioxide. Right? I taught you how to use the arrows, right? So I'm drawing like an outward arrow, releasing carbon dioxide. But during this process, we are going to completely break down the glucose, right? So because I'm going to release the carbon dioxide, right? So that means we have to release lots of energy. So the Krebs cycle is going to release 3 types of energies. Can you write down using the correct spot? Write down the 3 types of energies that are going to be released by this Krebs cycle. Can you complete these arrows? But remember, you are generating the energy, right? So write down the energy towards the correct side of the arrow. You are creating 2 ATPs again. You have to write down the number of ATP always. So the main energy is the ATP. I said there's 2 ATPs. And the 2 coenzymes, right? NADH and FADH2. Don't write FADH, you're losing a mark. Write down the unloaded versions. Is the Krebs cycle clear? So I'm going to go to the next page and write down what we did. So during the Krebs cycle, the pyruvate broke down into acetyl-CoA. And then acetyl-CoA further broke down into carbon dioxide. But this process released lots of energy, right? So energy is in the form of ATP. It's not an input, no? It's an output. So the energy needs to be in the form of ATP, NADH, and FADH2. I'm going to highlight this as the energy. So this is what we did. You can just look at this and write down the 4 inputs and 4 outputs. You can write down pyruvate or acetyl-CoA as the input, right? Or you can write down slash and mention both, whatever. Write down 4 inputs, 4 outputs. So I'm going to start with the outputs. I can see carbon dioxide. I can see the 3 energy sources. So what gave the carbon dioxide is you can say pyruvate, or you can say acetyl-CoA, or write down both like I do here. And then download it once. No need the numbers here. I'm not writing the ATP numbers. If you want, you can write. This is clear? Everyone finished? Pragati finished? Okay, so I'm going to just summarize whatever we have done so far. Right, so cellular respiration is breaking down glucose until it releases all the carbons, all the energy inside the glucose. Right, so I did glycolysis and then I did the Krebs cycle. Glycolysis is not inside the mitochondrion. Krebs cycle is inside the mitochondrion. It's in the matrix. So glycolysis, we break the glucose into pyruvates. You can add the number because you're splitting it up. It needs to have 2 pyruvates plus the energy. The next one is, I'm going to ignore the acetyl coenzyme A. We're taking pyruvate into the matrix. And inside the matrix, we're going to break down the pyruvates until it becomes carbon dioxide. Right? This process produces again energy in all 3 states. This is what I did so far. So this is the summary. You can just put this on the wall until you remember this. And I have little bit space for the electron transport chain. Can you highlight the ATPs so far? We have produced 4 ATPs altogether. Right, so far we have produced little bit of energy. 4 is very small. So when I started the cellular respiration, I said, I'm going to break down this 6-carbon compound until it becomes carbon dioxide. Right? So after the carbon dioxide is formed, you can't further break down the carbons, right? You can't break an element. Carbon is the final element. So the glycolysis, we broke the carbons until it becomes 3-carbon. And the Krebs cycle completely finished the carbon breakdown because we produced the 1-carbon carbon dioxide, right? But we have like one more stage to go, but we are not going to split the carbon anymore, right? So carbon splitting is completely finished. So the electron transport chain is not going to further break down any carbons, right? So electron transport chain is going to convert all the other source of energies. We have created lots of NADH during glycolysis, and we have created lots of NADH again, and FADH2 again, not again, FADH2 during the Krebs cycle. So NADH and FADH2, they are carrying energy, but this is not the usable type. So if in case we need to use the energy needs to be converted into ATP, right? So we're going to convert all the other coenzymes back to the energy, the usable energy, ATP, using electron transport chain. Right, so you have to know the main purpose of doing electron transport chain. You're not breaking down any carbons, right? So carbon breaking down is just completely finished. But electron transport chain is going to convert all the other energy sources, coenzymes, back to ATP. Let's do the electron transport chain. So I'm going to do the electron transport chain now. The third stage, you're going into the mitochondrial cristae. Is this clear? We're going to write down the steps of electron transport chain. Huh? If you want, you can draw this, but these are not the main steps, right? So write down the topic. You're doing electron transport chain. Write down the location. Don't abbreviate it. No? If you write ETC, no marks. Do not write this. Write down the location. So let's draw like a big part of cristae. Don't draw this. What I'm going to do is I'm going to enlarge the cristae. Don't draw this. I'm going to erase this. So shall we draw like a very big part of cristae? Like I'm going to draw part of the outer membrane and part of the inner membrane with the cristae. Yeah. So I'm going to enlarge this part here. Okay, before I enlarge, can you write down, can you draw the proteins inside the mitochondria? Let's go through the 4 proteins. There are proteins inside the mitochondria, right? So one example protein is something called cytochromes. you have to know cytochromes again in unit 4, right? So cytochrome is a protein complex. There are different types of cytochromes. You will look at cytochrome B, cytochrome C, these are complexes. But overall, let's take the cytochromes. So I'm going to highlight the cytochromes. Can you draw like cytochromes like this? This is just a representation. Just draw it like that. The red dots are the cytochromes. Right? So this is the cytochrome. This is another cytochrome. This is another. This is another. This is another. So one of the main proteins inside the mitochondria are the cytochromes. You have to include cytochromes when you're explaining the electron transport chain. Right, so when you're explaining the steps of electron transport chain, don't use like very complex steps. This is a very complex stage, but I'm going to do it. I'm going to give you like the stages, whatever you need for the VCE level. Don't include like somewhere in university, the first year or second year stuff, right? No need. But this is a very, very complex process, huh? So let's enlarge this part of this cristae. So I'm going to draw the outer membrane, inner membrane with the cristae, plus with some of the cytochromes. So cytochromes are proteins inside the mitochondria. Right? I need the space, so I'm going to erase this first. I'm going to enlarge the outer membrane, inner membrane with the cristae. Can you draw a couple of cristae? And then draw the cytochromes. Can you draw one cytochrome per cristae to make it simple? So the red circles are the proteins, huh? So you have to remember the protein as cytochromes. I'm not going to label it. Okay, leave the last one. I'm going to change it. So all the red ones, I drew cytochromes, 3 of them. So all I want you to remember is cytochromes as proteins. Cytochromes being proteins, they can be affected by, as an example, high temperature, pH changes, inhibitors, everything. Right? So that's why I'm highlighting the cytochromes being proteins. They are proteins, right? So if you have a protein, it can be inhibited by normal protein factors, right? Okay, and can you draw another protein at the end? So this is the protein, this is an enzyme. This is the enzyme that actually going to make the ATP. So the last protein we are using is an enzyme. This is the ATP making machine known as ATP synthase. Synthesize ATP. You see the A suffix means it needs to be an enzyme. Right? So I introduced 2 proteins inside the mitochondrion. You have cytochromes along the cristae, plus the ATP making machine is the enzyme ATP synthase. Can you write it down separately? The 2 proteins we are using here. We are using cytochromes and ATP synthase. right? So towards the end, when I give the questions, you need to know the enzyme, the protein types, right? So cytochromes, ATP synthase, include them in your answer, you get like full marks. Is this clear? The red circles are cytochromes. The green one is the ATP synthase, right? So hurry up, now let's do the steps of electron transport chain. Remember, this is a very complex process, so I'm just going to make it simple. Don't think too much. Huh? So the purpose of doing electron transport chain is, we have energy stored in these coenzymes, right? The main purpose of doing this is to grab the energy and convert it into ATP, right? So towards the end, you have to have ATP. We are creating ATPs from the two coenzymes. So I'm going to give you the steps now. I'm going to use 4 steps. The first step is, so we have created coenzymes throughout the Krebs cycle and the glycolysis. So we have created coenzymes throughout the glycolysis and Krebs cycle. So these are the sources. So all these coenzymes we have created so far going to come from the matrix towards the cristae. Right? So the coenzyme is going to come towards the cristae. And the number one is, the first point is, they're going to unload their high energy protons. So can you write down number one as unloading high energy protons? So coenzymes going to unload their high energy protons. So they become unloaded. So coenzymes produced throughout going to unload their high energy protons towards the cristae. Right? That's what I have drawn here. Coenzymes created throughout gonna come and unload their high energy protons towards the cristae. That is number one. Is the number one finished? Hurry up. Right? So just write down the unloading part, right? Okay, so after unloading these high energy protons, it's going to flow towards the ATP synthase. But when they are traveling, they need to go through the cristae, but they need to go through the cytochromes as well. Right? So you are now describing the flow of high energy protons. I'm just including the keywords. Can you include the high energy proton flow? But this flow needs to travel along the cristae through the cytochromes. Can you just move it like this? So I'm going along the cristae, but I'm going through the cytochromes until I meet my ATP synthase. So the flow of high energy protons will move along the cristae, but going through the cytochromes. Include the cristae and the cytochromes. That's it. And you need to have the flow of high energy protons. I'm just writing the keywords. Huh? Next one. Coenzymes unloaded their high energy protons and then they traveled along the crystal through the cytochromes. And finally, they will go through ATP synthase. That is number 3. Don't write number 3 yet, right? You can just draw your high energy flow through the ATP synthase, but I'm going to break this down. Leave number 3, I need to explain something. But remember I said this is a very complex process. I'm just making it like simple something. But if I say this is a high energy proton, let's assume like high energy proton has a energy component. And if I remove the energy part from the H+, I will end up with normal hydrogen. Just assume like this. So we are dealing with high energy protons. I'm going to separate out the energy component from the high energy proton. If I separate the energy, I will left with the normal hydrogen. Just write it down like this. So this is what happens during the ATP synthase. When the flow of high energy protons traveling through the ATP synthase is going to extract the energy from the H+, and it will convert the energy into ATP. Right? When the flow of high energy protons traveling through the ATP synthase, the energy component going to be extracted and convert into ATP. That's the number 3. Can you write down number 3? So flow of high energy protons travel through the ATP synthase, right? So that's what needs to be on this third point. But when the high energy flow travel through the ATP synthase, energy going to extracted and converted into ATP. This is the main purpose. We are producing ATP here. Write down the number of ATPs. We are producing the maximum 26 to 28 ATP. This is not the number you learned last year, but Bika changed the number recently. So this is the number you have to learn, right? Whatever the textbook says, it needs to be 26 to 28. Right? Is this clear? So when the high energy proton flow go through the ATP synthase, I'm going to extract the energy, and ATP synthase going to extract the energy, convert into ATP. Right? You have to give the number. The number has to be 26 to 28 ATP. It is very, very big compared to the glycolysis 2 ATPs or Krebs cycle 2 ATPs. We are producing a large number of ATPs. If something happened to electron transport chain, you're losing 26 to 28 ATP. That's a very big number. This is a significant number, right? Compared to the 2 ATPs previously produced in each stage, this is a very big. Is the third point clear? Okay, the last point, I'm going to erase everything to explain the last one. So I asked you to assume the high energy proton as the combination of the energy and the hydrogen, right? So from this, I extracted the ATP synthase, extracted the energy component and converted into 26 to 28 ATP, right? So now I'm left with the hydrogen. So the hydrogen that's going to leave the ATP synthase needs to accepted by the oxygen that we breathe in. Can you remember oxygen is an input of cellular respiration, right? So this is the place we are using the oxygen. So oxygen is going to go and grab this hydrogen. Hydrogen combined with oxygen producing water. Is this clear? High energy proton, the energy component, ATP synthase, extract the energy into 26 to 28 ATP. And whatever the remaining hydrogen going to be accepted by oxygen to create water. So this is the final product, water. Otherwise, we haven't used the oxygen yet, right? So this is why you have to breathe in oxygen to accept final hydrogen. So this is the number 4. All you have to write for number 4, this is very, very important. I see always students not writing this last point, you're losing at least 1 or 2 marks. 4th point is hydrogen that come out of this ATP synthase needs to be received by oxygen to create water. Is this clear? There are questions, right? We pass exams asking you to explain the electron transport chain. It could be 3-mark question, 4-mark question. The most important thing is you have to mention the creation of ATP. Plus, you must include this. This is something you might forget to write. You're losing a lot of marks. Just trust me. Right? You have to say hydrogen needs to be accepted by oxygen to create the final water. Right? Hydrogen accepted by oxygen. This is what you're going to breathe in. You're going to inhale oxygen. So your oxygen can go and grab the hydrogen to form water. So if you finish, can you write down 4 inputs and 4 outputs? I'm going to summarize this. So this is what we did in electron transport chain, right? Coenzymes went through the chain to produce ATP. You need to know the number 26 to 28 ATP. Plus we have oxygen to produce water. Right? So if you want, you can highlight the inputs, outputs. Oxygen becomes water. You can clearly see all the inputs, outputs. I need 4 inputs, 4 outputs. So the 2 main inputs are the coenzymes, right? NADH and FADH2. So we went through this electron transport chain. So we can remove the energy from the coenzymes to create ATP, right? ATP is an output. So this is the first case, you're having ATP and NADH, FADH2 on 2 different sides. And the last one is oxygen going to be the final proton hydrogen acceptor to produce water. Right? Now you can write down whatever the unloaded version. These are dot points. You must know the inputs, outputs. Huh? If you finish this, can you calculate the total number of ATP we produced so far? So this is different to the whatever you learned last year, right? Don't go through the last year's numbers. Anyone needs page? Can I go to the next one? Shania, you finish? I'm going to go to the next page. Write down the ATP produced in each and every reaction. How many ATPs in glycolysis? How many ATPs in Krebs cycle? How many ATPs in electron transport chain? Can you calculate the total? Total. It's just 30 to 32, yeah. But can you highlight the electron transport chain? So this is the reaction that's going to, this is the state that's going to supply the largest number of ATP. If something happened to electron transport chain, you are losing a significant number of ATP. Right? So the remaining ATPs can't sustain the live organism going to die. If something happened to this amount, organism, the cell gonna die. I want you to go with the numbers. Don't say electron transport chain produce a big number of ATP, right? So without saying a big number, just give the number, right? Electron transport chain is very important because it produces 26 to 28 ATP compared to the glycolysis 2 or Krebs cycles 2. Right? Right on the last term, we used electron transport chain, right? We used oxygen in the electron transport chain. So it was the final proton acceptor to produce water. So if something needs oxygen for whatever the reason, the entire stage becomes an aerobic process, right? Aerobic means a particular reaction stage requires oxygen as an input. So electron transport chain needs oxygen, so it can grab hydrogen. So I'm going to say electron transport chain is an aerobic process. I just need to introduce this term, right? Aerobic means, you learned this last year, it just means oxygen needed. Because electron transport chain needs oxygen to accept hydrogen, it must have oxygen present. So the entire electron transport chain is an aerobic process. If something happened to the electron transport chain, if this reaction stops, this is very, very important. Huh? I ran out of time, just give me 2 minutes, I'm going to finish this. If something happened to electron transport chain, you're going to interrupt the cycle of these FADH2 and NADH. So as an example, if you block the electron transport chain without having oxygen, so you're going to stop the entire electron transport chain, right? That means these coenzymes can't unload their high energy proton. That means Krebs cycle not getting the unloaded versions. Is this clear? If something had to happen to electron transport chain without oxygen, it's going to interfere the Krebs cycle as well. Because if the coenzymes can't unload their high energy protons, the Krebs cycle won't get the unloaded versions to grab their high energy proton. Right? So if the electron transport chain stops because of not having oxygen, Krebs cycle also going to stop. Can you write down Krebs cycle also? That means an aerobic process. Because Krebs cycle electron transport chain, they are interdependent. You can't just have the electron transport chain without the Krebs cycle, because, or you can't just have the Krebs cycle without the electron transport chain, because Krebs cycle need the unloaded coenzymes coming from this electron transport chain, right? So if the electron transport chain needs oxygen, Krebs cycle also indirectly going to need oxygen. All I wanted to write it down. Krebs cycle is aerobic. Electron transport chain also aerobic. But glycolysis, we did not use any oxygen, so it is an anaerobic process. This is something you have to know if you're doing the SACs or whatever. Remember, glycolysis is anaerobic. Krebs cycle, electron transport chain. Both reactions inside the mitochondrion, they need to have oxygen present. Both of them are connected, so both of them need oxygen. They are aerobic processes. Okay, so I ran out of time. Oh my God. I'm going to show you a question. You can do it when you have time, but I did everything for the question. This is the hardest question I have seen from 2002 to 2022. This has like a big concept behind this. So do it. I ran out of time. It's like half an hour. I'm going to show you the exam paper. I'm going to give the link. If you have time, just do it. It is very, very important. If you have a SAC, they always get like the idea concept from this question, right? Give me a minute. I'm going to give you the link. I have it in my email as well. If you want, I can email it. Okay, I'm going to share the screen. It's 2016. I'm going to send the link. It's 2016. Question 4. It's a very big short answer. It was 6 marks, right? If you don't know this concept, you lose 6 marks. 6 marks means a lot. So this is the question, right? So question 4, 2016. When you have time, please do this, right? It's a very big question worth 6 marks. 6 marks. To get the 6 marks, you need to get the entire row correct to get the 1 mark, right? So go through this slowly. The answers, you can find the answers online. Yeah. If you need the answers, I can drop the answers. I have the answers, but not the question. Here's the answer sheet, but read the question and do it properly. Yeah. Right? If you understand everything, you can leave. If you need to explain, like me to explain something, just stay after the class. I'm done. Bye.